Hello, students. For this lecture, we're going to talk about the material from the final unit, and we're going to start with that material. That includes the fungi and the animals. And so we'll look at a few earlier chapters before we get deep into animals and animal evolution. Of course, here we have a lion and her cub. There is also a lion's mane fungi, a mushroom that is really cool and delicious to eat. And when I was a much younger man, it was the first wild mushroom that I found in the wild that I could then bring home, check with my mushroom professor, and then brought it home and fried it up in a pan. And so it has a certain amount of excitement for me. But we'll focus on a variety of different life cycles and different things within, as well as the unicellular protozoans. In the same way that we included algae with the plants, we'll talk a little bit about the unicellular protozoans as well. So what are the protozoans? Proto meaning first, zoan referring to zoology, animals. These are unicellular single-celled organisms, often with all of the organelles and a fair amount of complexity. Their reproduction occurs via binary fission, usually. We talked a little bit about, in other lectures in lab, talked about cells sliding up next to each other and having some genetic exchange. But in terms of reproduction, in terms of the process of making more, they divide. It's a simple mitosis of sorts, but when it is utilized in a single-celled organism, it is called binary fission. So you're basically going from one cell to two, but in this case, the only thing in that cell, the only thing in that animal or that organism was that single cell. So that's a way of dividing and creating more. Of course, what's going to happen is after they go through all the stages of the cell cycle, they will all be identical to each other. How do they get their energy? Well, we talked, of course, about the plants and how they used photosynthesis. And of course, that's why we included the algae with that material. Well, the material in the protozoans are more heterotrophic. Hetero meaning from outside, and trophic referring to energy source. So it refers to their energy source, whereas we talked about the autotrophic photosynthetic organisms. Many are called phagocytic. So what happens is they'll draw something from the outside into a vacuole, use digestive enzymes to break it down, and then utilize those products of that digestion to help run the machinery and help them live in their source of energy. Usually, these protozoans are motile. They move around, and there's a variety of different ways. We talked a little bit about the cilia of the paramecium versus amoebas, which use pseudopods, which literally translates into fake feet, including Entamoeba histolytica, which causes amoebic dysentery. So this is also one of the reasons why the presence of a lot of amoeboids is why you make efforts to not drink the water directly from the tap when you're in certain countries outside of America. Obviously, in the situations, those that live there, they're kind of born with it. They develop a resistance to it or go through developing a resistance to it, and they can drink from the tap easily, but that is not the case for those that are visiting the country. You also have those with flagella, and we kind of looked at Euglena as sort of having a flagella. That was, of course, a flagellated, that was, of course, a flagellated algae, but in this case, other types of flagella are used for moving these protozoans around. Trypanosomes, which we'll see a picture of shortly, cause African sleeping sickness. Giardia is a very common zooflagellate. You find this in general in still water, and you will get it if you use that still water for you will suffer the consequences of Giardia if you drink water that has not been altered in some way or tested or sent through a filter or the like. My family, right when I was finishing up high school, my family went on a week-long vacation through the Boundary Waters up in Minnesota towards Lake Superior, and we just drank all of the lakes that we were swimming, that we were canoeing through and paddling through and camping, and sort of fortunately it waited until after we were back from the camping and in a hotel room in Minneapolis when the Giardia hit the whole family. And I think there were 4 of us and one bathroom in the hotel, and that was not enough. Anyway, just be careful of these. They can cause a lot of different complex problems. The final group are ones that don't move, the apicomplexans. Probably the most famous apicomplexan is the one that causes malaria. So it is a protozoan. You may hear about the Anopheles mosquito passing malaria between individuals and being the vector for it, but it is actually the apicomplexans that the Anopheles mosquitoes carries that actually and then injects into your body when they're slurking on your blood, getting a nice snack for themselves. Well, what happens is the apicomplexans then enter your bloodstream and can give you malaria. So a closer look at the paramecium, you can see that it very definitely has a lot of very important, interesting, specialized organelles. You can see that there is this contractovacule, which helps maintain turgidity. There is actually an oral groove where food can kind of come in, slide in through here, and be put into vacuoles, and the like. And of course, the cilia are found all along the outside, and those can pulse and beat. Those can pulse and beat as the protozoan goes through the environment. Other types of protozoans include the amoeba, and you can see the little pseudopods. In this case, this is a foraminifera. These are very interesting. They're found often by the sea, and they create these very interesting shell-like organisms. But of course, they're rarely seen at a concentration high enough to really just see in the water. But when you take a sample and when you filter it down, you can see these very interesting single-celled animals. And then in this case, the sporozoan mentioned, trypanosoma, African sleeping sickness, the malaria here, and the like. You can see how it has injected itself into the inner workings of a red blood cell. All very challenging diseases. So we often associate protozoans with these conditions or diseases that they cause. Now there's another unique group that are called slime molds. And these essentially, if we're going to say that algae are, if we're going to create a super analogy, an algae is to plants as protozoans are to animals, then slime molds are to the fungi kingdom. And so the protists, these unicellular organisms, this whole kingdom kind of has these relatives. But the fungi-like ones are very unique and different. They have the structure like fungi. They do recycle dead plant material. Oftentimes people will see water molds, which resemble true molds, which are actual fungi, but again, they are this separate group called the slime molds. And what's interesting is that in this case, a particular water mold that is historically significant is, of course, the potato blight of 19th century Ireland. So this was an agricultural pest that ruined large portions of the crops in 19th century Ireland, and had significant consequences for death of those that remained in Ireland, and of course the demographics of those that, say, came to America or sought refuge elsewhere, because potatoes had become, particularly for those that are poor, a significant part of the diet of the Irish people. So we used to kind of stick them in with the fungi, but the unique thing is that they have at least a stage where there is a flagella. And that is not the case for the fungi. The fungi are like, no flagella here. Nope. Move on. Go into your own group. So we kind of think of them together, but honestly, they're just these kind of strange exceptions to the biological world, but still very interesting. One type of slime mold is what are known as plasmodial slime molds. And the process of this is very much like a variety of other life cycles that we've seen, where you have a zygote, a diploid zygote, which will divide and divide, and out of those sporangium will occur, and those will go through meiosis to create spores, which will germinate into these amoeboid and flagellated cells. They have a very complicated life history, and one of the interesting things is when they're kind of in this stage, they send out a chemical, ADP. Well, they send out a chemical called AMP. It's a cyclical AMP, or CAMP. Cyclical adenosine monophosphate. And they send it out to the environment, and all of these like loose cells all come together to form this super network of plasmodium. And then there is the development of these reproductive structures, these spore-bearing structures, where meiosis occurs and the cycle continues. Fascinating things, except when they are on your sandwich. So that leaves us with the fungi, and that is the other non-animal group that we're going to talk about for this Unit 5. Sometimes it's easier to identify something with what they're not. They don't have chloroplasts and they can't photosynthesize. They're not animals. They are chemoheterotrophs. They're heterotrophic. They get their energy from other things. They get their energy from chemicals of other things. Just like you and I eat a sandwich, we're heterotrophic. And fungi are the same way. But of course, when we eat a sandwich, we stick it into our maw, we ingest it, and it goes through our digestive tract, versus fungi that kind of will grow into it. They'll actually release chemicals to break down the material externally, and then as that material breaks down, they will accumulate and take in the nutrients from the material that is being broken apart, and then continue growing and the like. Most of the fungal life histories and life cycles have spores. And those spores are found either during sexual or asexual aspects of the life cycle, depending on the organism that we're looking at. Ultimately, looking at a variety of characteristics, we have determined that, in fact, fungi may have more of a common ancestor with the animals than the plants, although traditionally they would be, like in a traditional class, they would perhaps be tucked with like a botany class or something like that. So, how are a fungus put together? What are they made of? Well, they're made of cells with cell walls, but the main structure of those cells tend to be in long, thin filaments, and these are called hyphae. They have a cell wall, but it's made up of chitin, which is a very different material from the cell wall of plants, which is, of course, made of cellulose. Now, you have seen hyphae. You have seen these single-stranded pieces of mushroom, these hyphae, in larger networks of material called mycelium. If you've ever looked at a mushroom, you may think like when you cut a mushroom, it looks like you're just simply cutting through solid tissue. But if you actually kind of look closely at the stalk of the mushroom, you can see that instead of just being a big solid tissue with 3 dimensions, what you're looking at is hundreds of these hyphae all intertwined and interacting. This is one of the reasons why, as they start to decompose themselves, it makes them very difficult to cut a mushroom. When they're nice and young and healthy, you can cut the mushroom very easily and see that fungal network. Mushrooms, at least the ones that we traditionally sell at a grocery store or eat, are a reproductive structure. But most of the life of these fungi tend to be in these large networks of hyphae called mycelium. And in the same way that, if you think about all the surface area that grows from, say, root hairs in a plant root, well, the mycelium give an incredible surface area for release of digestive materials, release of digestive chemicals, and the subsequent absorption of those materials into the hyphae. So it's an incredible amount of surface area which is there to do the decomposing. And you can see, of course, in this case, some mold on the side of not a water mold, not a slime mold, but a true mold found on the side of these strawberries. And when you look up close, you can see kind of speckled, the mycelium made up of hyphae are mostly white, but then there are these little black dots. And these aren't seeds from the strawberry. Instead, those are the reproductive structures. So we've taken the life cycle of a group of fungi called zygospore fungi and projected them onto a piece of toast. So, yum. But what we're looking at here is you can see a situation where you have a zygote, which creates this large structure. That's what those black dots were in that whole network of mycelium, then out of those black structures, cells will go through meiosis, creating haploid spores, which will then create all sorts of hyphae, which will create all sorts of mycelium. And these mycelium will also send out spores, creating asexual reproduction. But then there are different strains or lines of these structures. We don't really have like male and female. It's just different mating types. And what you can see is once you have the hyphae of two different mating types, the plus and the minus mating type, reach out to each other, well, this whole structure is haploid. And what happens is you have nuclear fusion. That is fusion of two nuclei, not the explosive energy of the sun or a nuclear bomb, but you have nuclear fusion here where the two nuclei will fuse, haploid, forming the diploid zygote, and the cycle continues. So again, this indicates there are two different mating types, and they can be asexually reproduced, or there can be a meeting of the two mating types to create a sexual process as well. The only part of this life cycle that is diploid is this dark black zygote, the zygospore. And that's what the ones are named of. So that's one major type of fungi. Another major type, or two types, are called the club and the sac fungi. And these are what we think of when we think of traditional mushrooms. The mushrooms themselves are simply the reproductive body. This creates haploid spores. So if you hold on to your portobellos for too long, they've got those gills that are all brown, and eventually the gills will start to kind of put a brown dust over things. Well, those are the spores being created. That is meiosis occurring right there at the very end. And the club fungi are ones where this sort of looks like a club, like it's trying to show here. Not like a boots and pants kind of club, but more of a club-shaped reproductive structure, whereas the sac fungi have more of a reproductive sac, if you will, where meiosis takes place. And most of the commercially available mushrooms, your white mushrooms, your portobellos and your baby bellows, these are mostly club fungi. But there are examples of a sac fungi, might be truffles, if you've ever heard of those or had the opportunity to have that as part of a meal, or truffle oil used from these very expensive sac fungi. So what are the benefits of fungi? Well, most fungi, as we've indicated, are what are called saprotrophs. They're responsible, they're heterotrophs. What kind of hetero? It occurs through the breakdown of plants, animals, and microbes. And this is extremely important in the process of nutrient cycling, so that there are nutrients available for the other plants, and so on and so forth up the food chain. So and there are even situations where you have they can break down nitrogen rich protein sources as well. We tend to think about them breaking down all sorts of things that have already passed or that are no longer living. But there are situations, there are specific types of mushrooms that will digest animals as a source of protein while they're still alive. Fungi are pretty famous for forming mutualistic relationships. We talked a little about those when we talked about the fungi in the roots. We'll talk about that in a second. Lichen are a good example. Lichen, of course, have color. They themselves, the body of this is all hyphae. The body of this is all fungal material, but the fungal material has photosynthetic algae within the overall structure. So in this case, it creates a little bit of an algae sandwich where you have fungal hyphae on the top, fungal hyphae on the bottom, and then algal cells living symbiotically. And again, there's exchange of nutrients and moisture and photosynthate and sugars from the friendly photosynthesizers. But lichens, if you've ever watched, they grow extremely slowly. And you can get an idea if you go to an old graveyard looking at a particular gravestone, you can kind of give an estimate as to how much growth it has had. You pretty much assume that when someone puts up their gravestone, it's not, they don't just take a lichen covered piece of something, but lichens will attach to it and slowly break down the materials of the gravesite, and of course ultimately are able to survive because they get energy from the algal cells living symbiotically. Another key fungal symbiosis that we talked about last time was the mycorrhizae fungi. These are the ones that will be able to perform nitrogen synthesis. They'll help supplement the these are sorry. We talked a little bit in the last lecture about mycorrhizae fungi, which actually form relationships with the roots and allow for breaking down of materials in the soil and making those available to the plants. So these really do help growth. And we even looked at photographs of plants with and without mycorrhizae. Of course, fungi are used in a variety of other ways. The origins, the discovery of penicillin was from a sac fungi called Penicillium. Yeast is a very small sac fungi. Usually the way we use it, it doesn't develop the sacs because we will use it in fermentation. But essentially, the yeast in those fermenting situations will actually develop alcohol as a waste product of their fermentation. And ironically, what will happen is they will take sugars in the environment, sugars, simple sugars, sugars the source of life, the source of cellular respiration, and break them down, but do so in a way that without oxygen, so there isn't oxygen inhaled. Yeast are, of course, important in the process of fermentation, both in bread and in alcoholic fermentation. In bread, they'll produce carbon dioxide bubbles that will add to the crumb or texture of the bread. And of course, in alcohol, they'll take sugars and perform cellular respiration, but they're unable to do it completely aerobically because in fermentation situations, they've removed all the oxygen from the environment, and as a result, they create this waste product of alcohol. And ironically, the yeast will simply keep creating alcohol until it is a level at which kills the yeast. So, if you, I'm sure there's a metaphor there for someone doing something with going through and using too much material. But for now, let's just simply say that these yeasts that we use in bread and in alcohol production, wine production, and in beer production are not going into the reproductive stage, even though, as I said before, these are sac fungi. And then, oh my goodness, nutritious, delicious mushrooms. I'm actually finally getting a couple in my garden, so I'm very happy about that. I've got a couple of mushroom gardens in my backyard, and they are so good to eat, delicious treat. Big fan of them, obviously, because I cultivate them, and the like. So, this slide, yay fungi. This slide, boo fungi. Because essentially, fungi can cause a lot of problems as well, particularly in terms of crop losses. And here we have, a gymnosperm, and you can see the fungus is kind of taken over, or the fungus have taken over here. This specifically is a type of fungus that is known as corn smut. And aside from just suggesting you use filters, if you ever look up corn smut on your internets, you can see the impact of the fungal infection of the corn into these large pieces. And one of the consequences of that fungus that creates this ergotized grain, or ergot, is that it includes within the structure, within the fungal infection, it includes what is essentially the key ingredient in LSD. And there are many that believe that ergotized grain, taking this and putting this corn with the rest of the corn, and then making some delicious corn pudding, because of the fungal byproducts in here, it ultimately led to people hallucinating, and some suggest that ergotized grain, corn, and other forms were actually the source of some of the madness surrounding the Salem witch trials. But again, we don't know, but it certainly is an intriguing premise. A second boo fungus is, of course, on grains, and can cause, amongst those that eat it, hysteria, convulsions, even death. You can see a variety of different fungal diseases, such as ringworm, athlete's foot on the outside, some that are deeper and spread throughout the body. With AIDS, when an individual has shifted from having HIV to the full acquired immune deficiency, that weakened immune system allows the immune system of the person suffering to be far more prone to fungal infections. And this is an example of another animal. This is a bat, which has this fungal, this white fungal nose, which is very indicative of a particular disease. And for our final boo fungus, this is a number of different ones. Candida albicans leads to yeast infections. It also can lead to what's called thrush. If you're on an inhaled steroid for asthma or that sort of thing, you need to make sure that after you do the inhaler, you will brush your teeth. Because what can happen is, while you're trying to send those steroids to help support your lung function, what can also happen is fungus just that are loose within the mouth and just all around us will in fact also enjoy those steroids and get energy and power and material from them, and that can lead to thrush. So if you're ever on one of those inhaled corticosteroids or and the like, you want to make sure that you're very conscientious about when and where you're taking your medicine. Ringworm, of course, who doesn't love a nasty skin infection to look at? Well, it isn't a ring. You can understand how when they first saw this particular structure, this particular piece of evidence that this person is diseased, well, it looks like a worm and it's in the shape of a ring, but actually it is in fact the fungus Taenia. All right. So the other thing that we want to talk about for this lecture and for the final lecture in the class are the materials from chapter 19, the evolution of animals. So animals are multicellular eukaryotes. So they're eukaryotic organisms, just like all the things we've been talking about since we first talked about the bacteria way back when. They are chemoheterotrophs. They're heterotrophic, and they get their energy from outside themselves versus inside themselves, and that is in the form of chemistry. So you may think, no, I eat ham. I'm a ham heterotroph. Well, you are enjoying and eating the chemicals in that ham, breaking them down and using them to build your own body, and using them to breaking them down to create energy for uses. As we kind of mentioned when we talked about fungi, animals actually ingest food and digest that material internally, while fungi, of course, release those chemicals, release the digestive enzymes into the world around them, and then slurp up the decomposed remains. Now, I'll try not to go too hard on this, but it is very interesting to me that when we think about food, when we think about our GI tract, essentially, we go through the process of eating the food, masticating, chewing, swallowing, and it goes through our stomachs, our intestines, and the like, large intestine, the colon, the whole kit and caboodle. Really, we're kind of, at least we are kind of this tube. We have this tube within us, this digestive tract, and the food kind of comes in and the poop goes out, but it is only that we are breaking it down and absorbing it. The food never really becomes part of us until we've broken it down to its constituent parts and absorbed those constituent parts. So that's something we'll kind of see, and in some of the more, I guess what we could call primitive animals, there isn't that level or that complete of a digestive system. So, unlike the animals that unlike the organisms we've talked about elsewhere, they have usually the method for reproduction is a sexual one. We don't have as easy binary fission or splitting apart or anything like that. Instead, the gonads go through a process of meiosis from a diploid organism to haploid sperm and egg. The sperm and egg fuse to form the diploid zygote, and the life cycle continues. Of course, part of that process is the development of the animal. what happens when it goes from 1 cell to 2 cell to 4 cell and the like. And here you can see sort of early, couple cells, 16 cells and the like, until you get more and more cells. And then eventually you have, in this case, more stages into the metamorphosis, in this case, of a tadpole, ultimately developing into a frog during that entire process of development. Now, when we think about it, and when we take our understanding of how development occurs, it provides some insight into how this may have evolved. Some of our hypotheses about how early animals would have developed. And the first step would be some sort of a colony. And you can think about the simple flagellate, but then ultimately multiple flagellates in an aggregated colony. You could then imagine that then ultimately there is that form sort of to create a larger thing, would create something, instead of just being a solid packed ball, to be more of a hollow sphere. And ultimately some of the cells within that colony would create specialization, and you'd have separation of different types of activities to give the this early animal, or give this colony some specialization and different tasks. We of course have seen this. We of course have seen this when we're talking about the Volvox, where you have certain cells within that large colony that create reproduction, and certain colony that do other things. The final step in this case, in terms of thinking about how a single flagellate to a colony to a large open sphere to specialization, the final stage is some sort of enfolding or creation of an invagination within that sphere, which ultimately creates sort of an inside and an outside of an organism. But while algae certainly has some key seaweeds and multicellular algae that are most likely to result on life on land, fungi perhaps are a bit more of a mystery, but we pretty much agree across the understanding of animal evolution that this originator was some sort of a protist, some sort of a protozoan, and multiples thereof. So there are, in fact, organisms that are kind of specialized protists that actually have similarities to this, what we're thinking about in terms of that multicellularity. And that is protists, unicells, all kind of working together in a colony. And there is a type of protist known as a choanoflagellate, which kind of represents that first stage. And these organisms are still around today. And of course, we have done analysis of the DNA to kind of see how those have shifted and how those are related to the animals as well, the ones that are here today. So how do we develop our evolutionary tree of animals? A lot of times when we're looking at it and talking about it, the way we describe it, it kind of makes sense in terms of accumulation of different characteristics. In the same way that the evolution of land plants gives us some insight into steps after step after step of going from an aquatic organism to a land plant, and the accumulation of characteristics that make it more and more adapted for land. We see similar sorts of things in the evolution of animals. The basic trend, and these are supported by, of course, molecular data, things that actually, when we look at sequences and how different organisms are related. We've done some incredible stuff with the Human Genome Project and then out to other organisms as well. And then, of course, developmental biology and seeing the consistent patterns in developing embryos that suggest a strong similarity and a strong common ancestry between different animals. So the main trends that we're going to see as we're going through the animal kingdom is, number one, multicellularity. We're looking at going from one cell to two cells to many cells. And ultimately, with that developmental folding in or invagination, we actually have the development of multiple germ layers. The ectoderm, which creates the skin, the mesoderm, which kind of creates the middle stuff, and the endoderm, which creates the innermost material of the digestive tract. Now, obviously, this is very different from plants, but you can certainly see similarities between the types of tissues and how these tissues develop within the plant kingdom and within the animal kingdom. We also take a look at the shapes and styles and look of the animal. There is a shift from radial symmetry. That is basically where you could, twist an organism around in any direction, cut it in half, and it would be symmetrical on both sides, versus bilateral symmetry, where there is basically only one axis where you can cut the animal in half and each of the sides look identical or are symmetrical with each other. Now, what good does bilateral symmetry, and why would there be a shift towards that? Well, when you have bilateral symmetry, you also have the capability of specialization of segments, specialization of areas, and you have the possibility of cephalization. You may have heard the term cephalus before, hydrocephaly. That has to do with the head. Once you have a head, you give that body or that animal some direction. You give that animal some structure and some separation and get a separation to different segments within the body where you have a lot of the nervous tissue towards the front and other parts of the body as you go further and further back. But the only time you can have a front and a back of an animal is when it's bilaterally symmetrical. Some simpler animals like the echinoderms and some others that we'll take a look at have more of a radial symmetry, which where's its head? Perhaps you've seen a lot of cartoon evidence in the form of one Patrick the Starfish that they do, but they just... are you sure that's where his eyes are? Because no, that's just the way he's drawn. Okay, protostome versus deuterostome. Now we're going back to our buddy, the stomata. the stoma, opening, proto meaning the first, deutero meaning the second, So when we're looking at this cartoon of enfolding, this area right here, this opening, will potentially, is known as the blastopore, and ultimately this will become the mouth of the organism, or the anus, as we enfold and create inward to create this tube within the body. Now, this shift from those that are protostomes, what this means is that initial enfolding point, that first opening that happens when material pushes itself inward during development, becomes the mouth. Ultimately, though, we see that more advanced organisms, in particular more complicated organisms, in particular the vertebrates, are deuterostomes, where that blastopore will actually be the anus instead of the mouth. Within those structures, we then can see the protostomes can be classified according to what kind of body cavity they have. They could be acoelomate, excuse my reading of that, acoelomate, which means they don't have any body cavity. They could be pseudocoelomates, which means that they have a pseudo or fake body cavity, or coelomates, those that actually have a true body cavity. And so when we're talking about those organisms that are protostomes, we will identify how their body plan is and how their presence of or absence of a body cavity can impact that particular organism. Deuterostomes, those organisms where the blastopore, that first opening, that first inward invagination becomes the anus, are all coelomates. And again, we'll highlight these things as we talk about some of the different components. The final trend that we see when we're considering the world of animals and animal evolution is the idea of segmentation. And that sort of takes the idea of bilateral symmetry and of cephalization and makes it much more fully formed, so that you have different parts of the body, different segments of the body, different repeating segments of the body lead to a variety of different specialization. And you may be saying, Eric, I'm no worm. I don't have segments like a juicy earthworm or something. Well, tell that to the repeating skeletal structures of your spinal cord and see how you feel about it then. All right. So, in terms of the overall evolutionary tree, we have a couple of animals that don't, in fact, have any formation of a stome. They don't have a full formation of a digestive tract using the endoderm, using the endoderm. So we have the sponges, which themselves, again, are kind of collections of different cells, these interesting colonies. We'll then talk about the cnidarians, the jellyfish, which have radial symmetry. They do, in fact, begin the process of forming a blastopore and forming an invagination to create the beginnings of an endoderm, the beginnings of a digestive system, but it is incomplete. And so as a result, you have material will flow into the mouth and be digested, but the mouth hole and the poop hole are the same for the jellyfish. So then the next main group are the protostomes, which include flatworms who have no body cavity versus those that have body cavities. And then you have the deuterostomes, where the blastopore becomes the anus. And the main groups there are the echinoderms and the chordates. But Eric, the quick-witted biology student, says, echinoderms have radial symmetry. If we're working our way up to more segmentation, more specialization, more cephalization, what's going with Patrick J. Starfish here? And I say, you are very observant, and all will become clear. So, symmetry, just kind of reinforcing this idea, the idea of radial symmetry. This would be how the organism is organized in a circular manner, and you can cut it in half in a variety of different ways. This is an example of a hydra, which is a jellyfish, a cnidarian, that is sessile. It doesn't swim around and ruin your day at the ocean. Instead, it is attached to something. But it is the same body plan. Food goes in, digestion occurs in the stomach in this gastrovascular cavity, and then any waste products go out the same opening. Bilateral symmetry, you, me, and our friend Turtle McGee here, it creates a body plan structure where there's a top, a bottom, a front, a back, and the like. Any other way that you try to cut this animal in half will not have symmetry on either side of the organism. There's only one way to cut this organism in half and have some form of symmetry, and that is bilaterally through the center of the organism. So again, reiterating our comparisons of the protostomes and deuterostomes. The protostomes are the flatworms, roundworms, mollusks, annelids, and arthropods, whereas the deuterostomes are the echinoderms and chordates. But it's important to note that the embryological development is what is considered when we are looking at that. So if you were to look at both the embryological development as well as the juvenile stage of echinoderms, you will see that they do in fact have a true body cavity. They do in fact have bilateral symmetry, and they do in fact have the second opening in their the second opening in their body during development will become the mouth, and the blastopore will become the anus, and that is shared with the chordates, which, as we'll talk about next time, are where we find the vertebrates. So in terms of development, the big stage is, of course, the forming of this invagination. And when this happens, what you can see is specialization of different cells. You've got the outer cells, which will be the ectoderm. You have the formation of these yellow cells that are actually forming this developing tube through the blastula, and those are yellow, and that's the endoderm. And then you have the mesoderm, the middle skin or the middle cells, kind of forming within and between the endoderm and the ectoderm. Ultimately, this imagination goes all the way through. The second opening here is going to be the anus. So you have a tube within a tube. You have a mouth. You have complete lining of that digestive tract with endoderm tissue. You've got the ectoderm on the outside in blue being all the skin, and then the mesoderm in red being all the good stuff, the guts and whatnot. Now, the question is, what happens when, particularly if you look at a cross-section, what happens in the mesoderm? Is there a body cavity? And we'll talk a little bit more about body cavities as we go. But here you can see an example of some of the most significant protostomes, such as the annelids and the arthropods, will have a true body cavity where there is separation, there is tissue, there is a body cavity that is completely surrounded by mesodermal cells, but protecting and found between the inner endoderm, digestive tract, and the outer ectoderm skin. Looking at that same process, of course, the blastopore here will in fact become the anus, and then as that endoderm forming occurs internally within, we will see the ultimate forming of, at the other end of this embryonic stage, forming the mouth. You still have, again, the same sort of idea, the formation of ectoderm, which will become part of the skin and the external structures, the endoderm, which will become the digestive tract, and then the mesoderm, which will become all of the rest of this body. And in this case, in the deuterostomes, all of them will have a true coelom where there is mesoderm cells surrounding the coelom, the body cavity itself. So what happens for those that don't have a true coelom? Well, for those that are acoelomate, that have no body cavity, there is no coelom, no body cavities formed within the mesoderm. Instead, this is solid tissue. And the example of this is, of course, the flatworms are acoelomate. This would impact their ability to move, their ability to adjust, their ability to get through the environment. They're not as bendy, and so the coelom offers the body cavity. The coelom offers that, but it also offers the not only does it allow for ease of movement, but it offers protection for the body goods, the body materials inside the body as well during movement and just in general. There is a group of roundworms that have a pseudocoelom. There is in fact a body cavity here, but what you can see is, unlike the body cavities that we looked at on the previous slide, the mesoderm doesn't surround either side of the body cavity. There's just the endoderm, this tube within a tube with a digestive tract. There's an opening, and then there's the mesoderm on the outside with the ectoderm forming the outermost layers of the embryonic animal. So as a result, this wiggly little worm can move around a lot easier, but it doesn't really offer the level of protection, and there is still very solid mesoderm outside, external to the pseudocoelom. So this is what a coelom, a true coelom, looks like. As I said before, it's continuous. The mesoderm is continuous with the endoderm, and then you have the coelom, the body cavity, surrounded by mesoderm all around, and then the ectoderm outside. So this allows for body movements. This soccer player is going to be able to stretch and move and the like because they're able to simply do that without squishing their innards, without squishing their organs. This allows space for the organs to become more complex. And there are, in fact, a few organisms that don't have a skeleton that are not going to be chordates, that are not going to have a skeleton inside. And in this case, the coelom further acts as a hydrostatic or fluid-filled skeleton. So it actually gives structure to the organism as well. Of course, for the soccer player, it is simply supplementing the skeleton itself. And the groups here, the mollusks, annelids, and arthropods. You can think about, as we talk about these organisms, how exactly this occurs in these, specifically with the hydrostatic fluid skeleton in some of these simpler coelomate animals. So the first main group that we're going to talk about are the sponges. These are characteristic animals of having multicellularity. They have these large bodies, sometimes small bodies that have these pores, and water flows through those pores, and there are different cell types within the animal that we'll see have a variety of different specialized function. They're mostly found, of course, aquatic, but they're mostly found in the marine environment. There are a few smaller freshwater sponges, but mostly we find them in the ocean. But the key here is that they are multicellular. They have a bunch of cells, but they don't have organized tissue. They don't actually have networks of cells or sheets of cells or tissues in any way actually acting. It's like almost a cooperative of all these different cell types working together. The way that these animals are structured is they have these flagellated cells called collar cells or choanocytes that line the inside of the sponge structure. And this is one of the reasons perhaps why we think of choanocytes when we think of the origins or the first protozoans to go multicellular. It's because we actually do see very specifically these collar cells in these animals. But what happens is they have these pores. Their name of their phylum is Porifera. They've got these pores, and water flows through those pores, and the collar cells are the ones that actually filter out the food, filter out material as it flows through, and allows it to take that food and then properly break it down and consume it. There are a variety of different reproductive methods. Mostly they do asexual, where they could just do by fragmentation. They basically break apart or break into pieces. And, if you take a sponge and plomp it in half, well, you go from having one sponge to two sponges. So it is a reproductive event of sorts. Looking at the opening of the sponge or osculum, you've got water flowing through all these pores, lined with these choanocytes, lined with these collar cells, which then take the material out of the water. And you have the amoeboid cells, which are kind of found crawling around within the sponge, delivering material throughout the sponge to the other cells. And then you have these other cells, which are dead at maturity, that are spicules that actually give the sponge its structure and gives it its three-dimensional structure. So you have the interaction. This is all from, they're all related. All these cells are from the same animal, but it's not like a true tissue. You just have these cells all kind of making their way within this large structure. A couple things. First of all, most of the loofah type sponges, they're not these. Loofah type sponges are from a relative of the cucumber or gourd. When you move away the plant tissue, that's those structural elements that provide that. If you were to use this as an actual sponge for your dishes, or as a in your bathing regimen, you would not be pleased with the decisions you've made. So we do actually have tissue-based animals, cnidarians, that, unlike the poriferans, actually have true tissues. They are, of course, radially symmetrical. For such a soft type of animal, it's impressive how much fossilized evidence. I actually have a few fossils of cnidarians myself. They have tentacles that are around the mouth with cnidocytes, and these cnidocytes are the barbs that they can use to attack their prey before they bring it into their gastrovascular cavity, before they bring it into their mouth. They may have toxins, and you think about even more complex, painful stings from the cnidarians. You could imagine that how much they hurt. If you've ever had a sting from a jellyfish, you can imagine how much that might hurt something that is smaller than you. They do, in fact, have two germ layers. So they have an invagination, they have a blastopore that becomes both the anus and the mouth, and that develops into an ectoderm, which creates the gastrovascular cavity on the inside. They have two germ layers, the ectoderm, which of course forms the outside, and the endoderm, which creates the gastrovascular cavity. There's a blastopore, an invagination, and that opening becomes, like I said, both the mouth and the anus for that animal. There are two different forms. There's the polyp, which is seen in anemones and hydra, and then the medusa, where the mouth is pointed downward, and that's what you see in the free-living jellyfish. So here we have a hydra. It has these tentacles. The mouth is right here. You can see that these tentacles have little cnidocytes. These tentacles have little cnidocytes that they can use to attack prey and then bring the food into the mouth. They are attached. They are sessile. And in this case, we have a bud happening here. You have an actual you actually have a asexual form of reproduction budding right off of the main body. Anemones, of course, as I mentioned, are another type of polyp, and here you can see all the tentacles on the outside, and perhaps you'll imagine a little clownfish looking for friends. You have, in the case of the Portuguese man-o-war, which is a far more complicated structure than it gives. It has the large tentacles and it has the mouth opening, but this large float actually helps it almost sail through its environment. And corals, of course, are another type of cnidarian. Those are a polyp as well. So these are tissue animals that have 2 layers. So the next step up, or the next thing that we're kind of considering about, are the flatworms, mollusks, and the annelids. And Lophotrochozoans. That is indeed a mouthful. Lophotrochozoans. And the flatworms are, like I said, in the next groups. They're still protostomes. The initial invagination becomes a mouth on these animals. The flatworms are the first group here. Planaria is an example of a flatworm that you'll find running around loose in nature. There are, as we'll see, a number of other more dangerous flatworms as well. So they have 3 full germ layers. Unlike the cnidarians that only have 2 germ layers, and unlike the poriferans, the sponges, that do not have any organized tissue and are just cellular structures, the flatworms have all 3 layers, but they do not have any body cavity. They do have the mesoderm will develop into a nervous system. They're bilaterally symmetrical, so they have cephalization, the formation of a head with nerve information, eye spots, and brain. And they're hermaphroditic. They have both male and female and will cross-fertilize in times of sexual reproduction. So here we have the cephalization, this end right here where there are eye spots and nerve tissue. They have an excretory system, so a kidney kind of spread out along the length of the animal. Like I said, they have cephalization, the formation of a brain, a concentration of nerves in one part of the animal, at one end of the animal, and the reproductive structures as well. In this case, both male parts and female parts. They have both. So the planarians are adorable, fun, cute, interesting to look at. If you actually cut them in certain ways, they will regenerate. So that is a form of reproduction. But there are a lot of flatworms, a lot of those in this group that have no body cavity and that are parasitic. And these include tapeworms. These are parasites, endoparasites. They don't sit on your outside, they sit on your inside. And they can grow from a few millimeters to upwards of 20 meters long within your body. And there is this organism attaches to the digestive system using these hooks in the head, and then have multiple repeating segments. Now, and what will happen is it will, one of the things about being parasite inside you, it's enjoying your digestive system. It's actually taking advantage. You're eating and then digesting the material, and this endoparasite will actually utilize the source of your, all your digested materials and take them away from you. Now, time was once, back in the 1920s, that tapeworm eggs were actually sold as diet pills. a more recent event of someone intentionally doing this to themselves. Many years ago, I worked at St. Norbert College, and there is a parasitologist there who would intentionally infect himself with tapeworms so that he had samples for his students. I would do a lot of things for my students, and I'm not sure I would do that. And I'm sure my students would do a lot of things for me, but I'm not sure that searching through my poop looking for tapeworm segments would necessarily be at the top of their list, per se. Another group of parasitic flatworms are the flukes. The flukes, like this one here, this is a blood fluke. It has again, both the male and female parts. It has the ability to attach. They're liver flukes, lung flukes, blood flukes, all named for where they inhabit. And probably one of the most famous types of flukes that is still quite common are the blood flukes, which lead to schistosomiasis. And looking at the life cycle of the schistosomiasis, humans are an important step, but there are other steps in that process. I think it was you need to kind of keep an eye out for other organisms within the life cycle of this parasite as it goes from one host to the next. And I think it was walking, if you were walking down the Nile, there was the potential for blood flukes in that water, so you needed to be very careful. So the first group of organisms that we're going to talk about that have a true coelom, that have a true body cavity, are called the mollusks. They have a complete digestive tract. They have a mouth and an anus. And the key things that all mollusks have, and there's an interesting diversity of them, are some sort of visceral mass of internal organs, this large muscular foot used for locomotion, and a structure, an organ called a mantle that envelops the visceral mass. One of the things that for many of the mollusks, the mantle will actually create the shell. So if we're thinking about things like a clam or a snail, the mantle creates the shells of those organisms and then lines the inside of those shells surrounding the visceral organs or the visceral mass inside. One other thing that we find in many is a structure called a radula, which is sort of a tongue-like projection near the mouth that has many teeth. Now, for instance, you may have heard of leeches, which are a variation or a type, relatives of the snail, but they're essentially a snail without the shell. They still would have a mantle that encloses or that envelops the body mass. That mantle, however, does not necessarily create a shell. So here's kind of an overall look at the body plan of a mollusk. You've got, in this case, a snail of some sort. You've got this visceral mass where all the body organs. You have a complete digestive tract from the mouth all the way down to the anus. You have this mantle, which is in this case creating the shell, the protective shell for it, but also helps protect the overall visceral mass. You have a large muscular foot. That's how snails kind of get through that world around them. And then at this end, at this terminal end of the digestive tract near the mouth, you have the radula with these scraping structures. And if you've ever watched a snail eat, you can see how they utilize the radula. Say you have algae on the inside of your aquarium, they may say, well, why don't you get a snail, and that will help with some of the algae in your aquarium. And you can actually watch it utilize the radula to scrape that algae off the walls of the aquarium and consume it. So there are a lot of different types of mollusks. One type are the gastropods. And gastro, of course, means stomach. Pod means foot. And the pod, of course, simply refers to the muscular foot. Gastro kind of refers to the main components of the visceral mass relate to the digestive system of this animal. Basically, it's a stomach and a foot. So gastropods include nudibranchs, who are also known as sea slugs, and you can see that here. During the larval stage, they'll have a shell, but that is then lost in adulthood. They still, of course, have the mantle, the foot, as well as other aquatic organisms like snails and conchs. They might be herbivores or carnivores. They might eat plants, or they might eat other animals. And in the terrestrial environment of a land snail, the mantle actually acts as part of the respiratory system as well. So these are the gastropods. Another group that is part of the mollusk diversity are the cephalopods. And whereas the gastropods are stomach foots, cephalus refers to a head. So we have head feet. These are octopuses, squids, and nautilus, like this interesting guy right here. A nautilus is a cephalopod that still, the mantle forms a shell. But it is not a snail. It is very much more related and has a lot more in common with the squid and the octopus. There's an incredible developed nervous system. In fact, a lot of research has shown octopuses to be amongst the smartest animals within the invertebrates, with complex sensory organisms and a large brain. And the relatives are not doing so bad themselves, but the octopus itself, though it is a favorite food, or if you're a hockey fan in Detroit, a favorite thing to throw on the ice, these animals have quite a bit more complexity and, again, intelligence than we hear about other organisms. The other main group that we want to talk about within the mollusks are the bivalves, which refers to the fact that they have two shells, two valves, like your clams, your clams, your oysters, scallops, and the like. Two parts of the shell. They still have nervous tissue, nervous cells, like these eyes that are photoreceptive. They are filter feeders, and so they send water through the organism and filter out any food that is found within. And that is how they get their food there. That is how they get their energy. The next main group of animals are the annelids. These are your segmented worms. And the segmentation is perhaps most obvious in this earthworm right here, because you can actually see these repeated segments found along the entirety of the body. It has a true coelom. It is a fluid-filled coelom, So it utilizes that coelom as part of the structure as part of its internal aquatic or hydrostatic skeleton internally. And this gives the animal something to push against and pull against so it can utilize its muscles to get through its environment. They have a complete digestive tract, a mouth at one end, cephalization, a brain, several hearts, and all the way down through the other end to the anus. Oftentimes there are specializations. The crop and gizzard that we find in most earthworms help break material down. Earthworms are known as sort of going through the soil and breaking it apart and loosening it and getting material from it and making it better for plants. They do have an entire closed circulatory system. And as I mentioned before, there is a brain here, and along the underside of the animal, there is a nerve cord going down the length of the animal. So here's a look at this animal a little bit closer. You can see the circulatory system has both dorsal and ventral blood vessels. The kidney system or excretory system has these structures found throughout these nephridium. So it's not just a concentration of kidney cells. Instead, it is these nephridium that go along the entirety. So they remove waste products via excretory pores. So they don't have sort of a single place where they urinate from. They just sort of urinate from pores all up and down the entirety of their body. There is this nerve cord that is found on the ventral side, on the stomach side of it. There is, of course, the true coelom. There is the digestive tract and musculature around it, which it utilizes to get around in the world. And you have two different muscular patterns, which is kind of cool. One that goes lengthwise, the other that goes crosswise. And so if you've ever spent the day watching a worm walk around, you can see how it kind of stretches and then contracts, and stretches and then contracts. And that is how it gets through the world around it. But it has nerve tissues, it has cephalization and a brain. But yes, it's indicated by these longitudinal muscles that go the length, and these circular muscles, which are more pinching. And these two different muscle types work against each other and both push against the fluid-filled coelom, which creates this hydrostatic skeleton. So, as I said before, the earthworm is probably the most well-known of the annelids. Other annelids include polychaetes, which are generally marine worms, including these Christmas tree worms and the clamworm. These are called polychaetes. They have these little structures, these setae, found on every segment. So they not only have segmentation, but they have specialization of those segments. Now, the Christmas tree worms are pretty cool. If you're swimming and you go up to one, they kind of hide back into their little hole. But again, these structures are setae that are coming off of these segments of this worm. There are also, there are the oligochaetes, which are the earthworms, and do not have as many setae. You have to kind of look a lot closer to see them. And then the third group of annelids are the leeches. And leeches have no setae whatsoever, still have the segments, found mostly in freshwater. Some are free living, but they do tend to attach to hosts where they perform sucker activity. And what they do is they essentially they have these sharp little teeth at the end of their bodies, and they use them to dig into the host's body and start slurking on their blood. Fluid feeders, in the fluids of the host. And they have two things. They have an anticoagulant, mentioned down here, Hiriaden, but they also have within their mouth, their mouth with these sharp teeth, they also have anesthetics located within their saliva, so that when they do bite you with those sharp teeth and they are slurking away, they have anticoagulants to prevent you from healing up so they can keep slurking on you for their meal, but they also have, so not only will you continue bleeding into the animal, which will enjoy your fluids, but their saliva has an anesthetic that makes it so you don't actually feel when they're biting into you and when they're on you, or when they're biting into you or slurking on you. So these characteristics are that of a leech that can really do quite well in terms of getting its food out of a host. Now, one of the things to consider is that historically, leeches were used for, quote, blood diseases back when we kind of sort of had the major humors, and that was the methodology in medicine. And obviously, we kind of look back on that and scoff, but what is interesting is that because of some of those same properties, we have actually incorporated leeches into some medicinal and healing practices. So these are the annelids, the last group that we're going to talk about tonight, and we'll cover the remainder of the material from chapter 19 in class next time.